the end, so it will explain that there is 5 minutes. And there is this bell here that then we'll use if ever they go too far, okay? It's not because to be cruel with them, it's just to give to everyone the opportunity to talk. And also I want to give to everyone the opportunity to question if ever you have some question, because I believe they are very clear. But at the end, sometimes we have questions or opinions. For the opinions, I will ask you to keep them for a discussion, general discussion. We should have time at the end for a general discussion.
Zebrafish embryos were used to capture the essence of the life force of movement. In microscopic studies of embryonic growth, visual distortions, physical vibrations, shadows, and microbial parasites randomly appear. The work showed how physical contact and looking creates reactive and deadly tremors. It showed that the embryonic organism is fragile and frequently dies in the process of research. Zebrafish belong to a group of model organisms. The processes of birth, fertility, and death are mirrored in the circulation and developmental regulation of our blood cells, a beautifully cohesive system whereby multiple types of cells with differing lifespans and functions are renewed throughout our lives from stem cells in our bone marrow. The maintenance, renewal, and division of blood is a continuation of the cell division process by which the structure of blood first came into being. The vascular and blood system generates the running current, bringing oxygen to our cells.
I'm going to talk about some of the ethical issues. I was in a bit of a debate whether to talk about the technology of magnetic resonance microscopy or some of the social and ethical, and I've chosen the social and ethical. What status should be given to newly created artificial life form? What about a life form that has been mutated, altered, hybridized, or created through transgenic manipulations? I would like to consider how the status of new life forms is inferred when the new life forms are variously considered to be either property, what if it's tissue, what if it's considered to be a life, or a cure, what about if it's an organism, or considered a member of society, or something alien? I will suggest that the language we use to describe the life we create and the images we use to depict it has a profound impact on the status that society will eventually ascribe to that life form. And it is therefore important that we choose our depictions and our descriptive language carefully. So let me suggest some titles. The word embryo, what's implied by that word? What do we imply if we use the word baby instead? What if we use the word zygote? How does that change the implication? Or the word child? What about specimen? Certainly these all apply to an embryo. What about the name Emily? What if we refer to it as tissue? Or as a patient? And certainly embryos are patients these days with the in utero surgery that takes place. By the way, these patients have their own doctors and lawyers nowadays because of the technology that where we've advanced. A very different kind of an interpretation. What if we simply call them life forms? Here we're calling them extremophiles and life forms. What does that mean? What about person? So again, the nomenclature that we use is very significant. The language used to describe extreme life forms will project attitudes and assumptions onto those forms, just as these names have done in the life entities involved in stem cell research and in cloning. So let's think about it. We're creating unusual new life forms now. We are going to be able to exhibit certain behaviors towards them. What's the list of these possible behaviors? They're really rather finite when you think about it. There are not many of them. We can do nothing. We can just simply let happen what is going to happen. That's one possibility. What if we exert our energies to bring the life into existence? We create them. They would not be created unless we took that energy and did it ourselves. Another option. Another behavior, we can nurture the life, however, within its own system. So we could provide nutrients or a more appropriate environment, but keeping it in its own system. What about intervening to assist the development and perhaps taking it out of its own system? We can suspend development. This is happening in human embryos where we freeze them. What's the consequence of freezing or suspending development of something and then bringing it about later? What about terminating it? We can terminate development or life. We can use life forms that we create for our self-interest. Perhaps as artists, we want to become known and have a name out there, and so we could use these in order to gain some notoriety. Or we could use them, life forms, for the sake of other people, in other people's self-interest, perhaps with no harm to the life form itself. How is that different from using the life form in the interest of others, but with the possibility of harming the life or terminating it?
One of the most important questions that we're going to have to entertain is actually comes out of something like that, right? Where we're going to have to ask, do we have the capacity to love the creatures that we create?
The next presenter is Sophia Roosth, and she changed the title for Screaming Yeast. So it's up to you now.
Yeah, I didn't just change the title, I changed the talk. So this is called Screaming Yeast, Sonocytology, Cytoplasmic Milieu, and Cellular Subjectivities. Saccharomyces cerevisiae, more commonly known as yeast, is a unicellular fungus with a cell cycle similar to that of humans. The first eukaryote to have its genome sequenced and a standard model organism in biology research, yeast is an organism that lends itself easily to multisensory experiences. It has been imaged extensively with light and atomic force microscopy. Sonocytology, a recently developed technique in nanotechnology research, uses a scanning probe microscope to record the vibrational movements of cell walls and amplifies those vibrations so that humans can hear them. Yeast cells vibrate at a frequency of about 1,000 times per second, and most cells vibrate within the frequency, though not amplitude, range of human hearing. Humans can hear a sound, any vibration that has a frequency in the range of 20 to 20,000 vibrations per second, or hertz. By amplifying the vibrations of cells, researchers essentially turn up the volume on cells. In 2004, Jim Janczewski, a scientist in the Department of Chemistry and Biochemistry at UCLA, with his graduate student Andrew Pelling, used an atomic force microscope, or AFM, to record the nanomechanical motion of yeast cells. And I think Jim is in the room today, is he not?